So my first step of the signal transduction pathway isn't necessarily a step, but it's just showing how the whole system is inactive, basically. So as you can see, the enzyme's inactive, the relay molecule, the protein kinases, and the transcription factor and the proteins are all inactive, just because that first reaction isn't happening between the G protein coupled receptor and the ligand. So it's basically just showing that this first reaction is needed in order to create more reactions, which activates the whole system, basically. So once that ligand actually attaches to the receptor, the G protein of the receptor, its GDP, is going to be replaced with GTP. And basically what this replacement does, it transfers the G protein to the enzyme that was once inactive before, and this transfer activates the enzyme. And this reaction that's happening right here creates a cellular response. And this response is basically the activation of the relay molecule, which was also inactive before right here. And then this active relay molecule is going to go on to activate the phosphorylation cascade. But first, before we get to the phosphorylation cascade, once this GTP is used up by the enzyme is going to become inactive again. So the G protein is going to be disconnected from this enzyme, and it's going to get its GDP back, and it's going to go back to the receptor as the ligand detaches from the receptor, so it can receive the GTP once again to repeat this cycle. So what this active relay molecule goes on to do is create a reaction between it and the first inactive protein kinase. And what that's going to do is create the first active protein kinase. And this activation is going to convert ATP into ADP, which releases a phosphate group, which is going to interact with the second inactive protein kinase, making it into the second active protein kinase. And basically the same thing is going to happen here, the conversion of ATP into ADP, releasing a phosphate group, interacting with this inactive protein kinase, making the third active protein kinase. And over here, it's basically the same thing, but just with different products and reactants. So ATP and ADP is converted as the inactive is converted into the active. So the phosphate group is released and it reacts with this inactive protein over there that was once here in order to activate the protein. And what that active protein goes on to do is it'll actually enter the nucleus in order to activate the transcription factor that was once inactive before, making it active. And that active transcription factor is going to go on to actually sit on the DNA that's located in the nucleus. And this is going to just trigger a reaction, making mRNA in the process. And then the mRNA is then going to exit the nucleus, going into the cytosol. And the mRNA is just going to be basically synthesized by a lot of reactions, which in the end will make a protein, which is the final product of the signal transduction pathway.